light we see, the visual light, is part of the electromagnetic spectrum. The spectrum is made up of radiation that has very long wavelengths, like radio waves, and very short wavelengths, like X-rays. The radiation we see we call color. Our eyes have evolved to see light between 400 to 700 nanometers, about the size of a bacterium. As light passes through the cornea, it is refracted towards the lens of the eye. It converges. The lens refracts the light even more to create a focal point on the retina at the back of the eye. Light coming from a source is traveling out in every direction. However, light rays coming from faraway objects or source seem to be parallel from the observer's point of view. Rays that flare out or diverge are more easily recognized when an object or source is closer to the eye. Myopia is when the eyeball is too long or the cornea is too rounded. Rays coming from far away cannot be focused on the back of the eye. The lens cannot flatten enough to focus the light rays on the retina. Myopia is corrected with a concave lens that causes the rays to diverge. The rays appear as though they are coming from a near object, and the focal point is now on the back of the eye. When we look at closer objects, the lens gets rounder to move the focal point from behind the eye to the retina. Hyperopia is when the eyeball is too short or the cornea is too flat. The eye lens cannot get round enough to bring the focal point to focus on the retina. Hyperopia is corrected with a convex lens that converges the light rays even more. MIT has created an illusion that could let you know if you need glasses. From far away, you should see Marilyn Monroe in this picture. As the picture gets bigger and closer to you, you should be able to see Albert Einstein. If it still looks like Marilyn Monroe up close, you may need glasses. We have millions of light-sensitive cells inside our eyes covering the retina. Some are shaped like rods, some are shaped like cones. They process the light into nerve impulses and pass them along to the cortex of the brain via the optic nerve. Rods are mostly concentrated around the edge of the retina. Rods transmit black and white information to the brain. As rods are more sensitive to dim light than cones, you lose most color vision in dusky light and your peripheral vision is less colorful. Cones are concentrated in the middle of the retina. There are 3 types of cone-shaped cells, each sensitive to long, red, medium, green, or short, blue wavelengths of light. Sunlight is colorless or white light. When we see the color of an object, it is the light that is reflected. A red guitar is absorbing all wavelengths except red. Red is being reflected back to us. It stimulates our red cones in the eye. They send a signal to the brain and we see red. We see yellow because yellow wavelengths stimulate the red and green cones such that the brain interprets the wavelengths as being in between red and green on the spectrum. How do we see magenta? It is not on the visible light spectrum, but we do see it. Magenta comes from red and blue light. So the red cones are stimulated and the blue cones are stimulated, but the green cones are not. So what is between red and blue on the visible light spectrum? It is green, but your green cones are not firing. Your brain invents a color, magenta, which is really the absence of green. Therefore, our brain is easily tricked when it comes to color. TVs use this phenomenon to generate all the colors we see with only 3 colors of light. When you see yellow on TV or on your computer screen, what you're actually seeing are red light waves and green light waves together. Your brain interprets this as yellow light. Remember, our eyes and brain do not directly measure wavelengths of light. We just have cones sensitive to 3 different parts of the visual spectrum. You can't mix color in physics. You can't mix a blue photon with a green photon and get a yellow photon. Color mixing is biological. What happens when our cones do not work properly? Color blindness is often inherited from the mother as the genes for making red and green cones are found on the X chromosome. 1 in 12 men are affected, 1 in 200 women are affected. Women need faulty genes from both parents. If you are colorblind, it means the instructions for the development of your cone cells are faulty, and the cone cells might be missing or less sensitive to the light. Or it may be that the pathway from your cone cells to your brain has not developed correctly. Color blindness is not all or nothing. It is a gradient from moderate to severe. How do you describe what you are seeing if you can't see colors other people see? You can liken it to tone deafness. The keys on the keyboards could represent the range of colors. Someone who is tone deaf will not be able to distinguish between a C and a C sharp. Being red-green colorblind does not mean sufferers mix up red and green. It means they mix up all colors which have some red or green as part of the whole color. Color is funny. It is something humans perceive. It doesn't really exist.